And we are back for yet another lecture. This one is going to be about aquatic environments. So let's first focus on the lake environment. One of the big differences between lake environments and soil environments or terrestrial environments is they're nutrient limited. There's much less organic material in a lake. Most organics are available as polymers, and they're degraded by extracellular or periplasmic enzymes. Often, phosphorus and nitrogen are limiting for growth in these environments, and the nutrient levels will dictate the microbial population in the lake. You can classify lakes by the type of nutrients they have. An oligotrophic lake, such as Lundy Lake shown here, has low nutrients, clear water, and very low primary productivity. There's not enough going on. And it's even if you have a primary producer, a cyanobacteria, that cyanobacteria is still going to use nitrogen and phosphorus, and if it's at low concentrations in the lake, you're not going to get a lot of primary production. A mesotrophic lake has an intermediate level of productivity, and a eutrophic lake has high biological activity. And here is a eutrophic pond shown here, and you can see it looks kind of scummy, etc. It has high biological activity. There's excessive nutrients, especially phosphorus and nitrogen. Another important thing about lakes is lake mixing, especially in temperate climates where you have big swings in the air temperature. In different times, you will get the whole lake turning around, and the biggest turn happens in the fall. When the temperatures get near 4 degrees centigrade, the top of the lake will cool. That water will be more dense. It will then sink to the bottom. As it sinks, it turns up the water below it, and you'll get this mixing effect. It's very important for these lakes because every year it mixes the nutrients. In the winter, the lake will then freeze. You'll get ice over the top, and you get very little mixing. There will be a less violent mixing in the spring. And again, these are very important because it mixes the nutrients of the lake. Okay, now right away we're going to have a think, pair, share. An example on a producer in a primary lake is what? What do you think the primary producers are? 5. When do you think the microbial activity of the lake will be the most active? Spring, summer, fall, or winter? There can be a number of different primary producers in lakes. Understand there's going to be some runoff from the ground around the lake. So actually trees and grasses and stuff do contribute to the amount of carbon that's in a lake. There's also aquatic plants, but there's also cyanobacteria. When is the microbial activity the highest? In the summer, when the temperature is the highest. Microbes have a central role in aquatic environments. They are major important primary producers. Yes, you get some contribution from aquatic plants. Yes, you get contributions from the land around the lake. But the bigger the lake is, the more important the primary producers, purple bacteria, green bacteria, and cyanobacteria are. They create organic carbon and they help fuel the food web. The main degraders are, again, microorganisms, and they will mineralize the organic compounds. Microbes drive the chemical fluxes, and they affect water quality. If there's too many nutrients, microbes will be there to take up all the oxygen, and that can be a problem. But they can also be very important in cleaning up water systems and removing excess carbon and making them usable again. So the common phyla that you see are shown here. These are done either by amplicon sequencing or by other methods of sequencing, metagenomics. And what you find is that there's about 21 phyla that are often found in lakes and 5 major ones: beta proteobacteria, alpha proteobacteria, bacteroidetes, cyanobacteria, and actinobacteria. And as you can see, actinobacteria are major players. One of the major players that you may be familiar with that's actinobacteria are the streptomyces, but it turns out they're not a major player in these environments. The actinobacteria that are major players haven't been yet cultured well. They are AC1 is the designation they've been given, and they can be up to 50% of the biomass of a lake environment. And this is something that you often see in lots of environments. You'll see a couple to maybe half a dozen major players and then a long tail of different organisms that are in this environment. Actinobacteria are single-celled microorganisms. They tend to be pretty small. In an amazing experiment, Professor Katrina McMahon and her research group, she is a professor at the University of Wisconsin-Madison, they actually isolated a single organism and sequenced its individual genome and were able to figure out what this organism's genome contains. It's small, it's about 1.2 megabase pairs. It has a low GC ratio. And Actinobacteria is one of the smallest genomes of free-living organisms ever sequenced. You can see here is a graph of G plus C content versus size of the genome in megabase pairs, and it compares a whole bunch of other organisms to AC1. And as you can see, it's a very tiny genome. So looking at the sequence, you can then determine the proteins that are there, figure out what all the open reading frames are, figure out what they do, and try to start making some guesses on the physiology of this organism. The things that you learn by sequence gazing are, it can metabolize xylose and ribose, it appears. It seems to be able to degrade polysaccharides like chitin. It may have carbon fixation, which is interesting. And it has many repair mechanisms for its genome. Finally, it has an interesting molecule called actinorhodopsin. And this is actively being worked on in Professor Katrina Forrest's laboratory at the University of Wisconsin-Madison in the microbiology department. So let's look a little bit at the chitin degradation. What chitin is long polymer chains of N-acetylglucosamine. And you can either break these apart into their individual N-acetylglucosamine units and then remove the acetylglucose and the amine and then go on to fructose 6-phosphate, or you can move the acetyl groups first and then break it into glucosamine, and then you can see this shuttles right into glycolysis. It turns out a number of different organisms are able to degrade chitin, and AC1 happens to be one of them. Another thing that was very interesting is the presence of actinorhodopsin, and this is a molecule that has a very close homology to bacteriorhodopsin. And remember, we ran across bacteriorhodopsin when we talked about a light-dependent phototrophy that wasn't photosynthesis. Bacteriorhodopsin is expressed under oxygen-limiting conditions for this microbe, and the microbe that we're talking about here is Halobacterium. This hydrophobic barrel-shaped protein spans the membrane and contains at its center a molecule of retinal. Retinal is a long carbon molecule with many double bonds and a nitrogen atom at its end. The double bonds allow the molecule to absorb light, and the nitrogen on its end can accept and release protons. Retinal is normally in a straight conformation, with the nitrogen atom facing the cytoplasm, from which it picks up a proton. Okay, so that's how it's normally sitting. Now, when light hits it, it changes conformation, and that N switches from a all-trans retinal to a cis retinal, and it moves where that proton is. It now goes from an acidic environment to a more basic environment, and it deprotonates. So when light hits it, structure changes, it flips over, the hydrogen is now deprotonated, and it leaves the cell. If you look at the structure of bacteriorhodopsin and you look at where that base is, and then the proton comes in here, light hits it, it switches, and then it goes out. And then it relaxes back to the trans state. And this happens over and over again, and it serves as a proton pump. It's thought that AC1 has the same protein, which they call actinorhodopsin, and it's one of the reasons it can dominate the environment that it's in, because it has this way of generating extra energy with this proton pump. Another group of organisms that you find in these environments that are prevalent are cyanobacteria. These contain plant-like photosynthesis, as we talked about before. Typical species includes the one listed here. In the spring in eutrophic freshwater lakes, they typically get a cyanobacterial bloom. The extent of that bloom is directly related to how much nitrogen and phosphorus is in the water. When this bloom crashes Other groups increase to degrade them, and one of the examples is the flavobacteria. All right, so let's talk a little bit about Anabaena. So is one example of a cyanobacteria that's found in lakes. It's a filamentous cyanobacteria. It's an obligate photoautotroph. Under low nitrogen conditions, it will actually make a heterocyst, and this is a specialized structure that allows it to fix nitrogen and photosynthesize at the same time, and we've talked about that. The typical strain is PCC 7120. It has a 6.4 megabase pair chromosome in a single circular fashion and 6 plasmids. So, a concept check. An example of a primary producer in a lake is, and which of these can take wood and break it down? And this makes you think about what you learned last time. All right, on to oceans. Oceans are different than lakes. First of all, they're a lot bigger. What that means is the contribution of land is negligible to most oceans. The water density is dependent upon temperature, and because the ocean can span all the way from the equator all the way up to the poles, it is a driver of ocean circulation. The pH of the ocean is about 8, and it contains a lot more ions. The 85% of the solvents in the ocean are sodium chloride, but there's also sulfate, magnesium, calcium, potassium bicarbonate, bromide, borate, strontium fluoride, and others. These are, washed off as the water runs across terrestrial lands, eventually ends up in the ocean. When the water evaporates, the ions stay there. So there's a lot more ions in the ocean chemistry, and that will affect what can grow. Ocean chemistry and gases. You have carbon dioxide. You have oxygen in the top 20 meters, and it's totally gone by 500 meters. It is again present at the floor, and this is because of ocean downwelling. If you look at the ocean surface, and at the ends are the poles, the middle is the equator, you will have these areas where the ocean cools, and then there's downwelling, and this pushes oxygen and gases all the way to the bottom of the ocean floor, and then it will just slide around down there in the currents at the bottom of the ocean, and then will upwell in divergent zones. There isn't a lot of metabolic activity in most places at the bottom of the ocean, so you actually see oxygen there. Nitrogen gas is present, and there is more gas of all these types of gases in cold water. The ocean also has light. There is a euphotic zone. This is the zone where light penetrates the surface of the ocean all the way down to where 1% of light remains. It depends on the amount of particulates, and it can be anywhere from 3 to 150 meters. This light will be used by organisms in the ocean. Who do you think will be the primary producers in the oceans? Once we developed the modern tools of microbial ecology, it was then time to start exploring the oceans using them. Scientists created a ship called the Saucer II, and the Saucer II was then used to sample the ocean. You can see in all the different places where they sampled, right? All the way in the Atlantic, all the way through the Pacific Ocean and around the Galapagos Islands. As they did this, they took the ocean water, collected it on filters, they captured took all the microbes off these filters, they lysed them, and then they sequenced all the DNA, doing metagenomics. Afterwards, they assembled this into large contiguous pieces of DNA, and they could make a lot of discoveries. Much of the phylogeny was initially based on 16S ribosomal RNA sequences, and it's really important that we take a little side note and think about that for a second. So if you're just looking at DNA sequence, how do you define a species? The classical way is looking at various characteristics, the morphology of the organism, looking at the different things it can do metabolically, and using that to organize and classify microbes. You can't do that with sequence, and so what they found is there's somewhat of a correlation between a 95 to 99% identity in sequence and being the same species. So yeah, there's a range, but that's where people are at. Genera have about a 92 to 96% identity. Okay, as scientists do this work and they decide, okay, everything above 97% we're going to bin into the same group, they will call that an operational taxonomic unit. They will sometimes assign species names to these OTUs, but understand that's kind of an approximation because all we're looking at is DNA sequence. If you look at the microbial genera, so you look at the analysis that's done over the whole ocean and you accumulate all that up and you look at it and then you look at the most common genera that you find, what you find are 5 genera, and then we're seeing that same pattern. There's this group of organisms that make up a large portion of what's there, and then there's everything else. And what you see is Polygobacter, Prochlorococcus, and Syncococcus make up a significant amount of the organisms that you find in the ocean. So those are the ones that we're going to talk about. Polygobacter was previously called SAR11 before it was isolated. Giovanni's lab since isolated that organism in 2002, and here's what they found. The species name is Polygobacter ubeki. It is an oligotroph that grows on low nutrient densities, and that may be one of the reasons previously it was difficult to isolate. It has slow growth. At 15 degrees is optimum growth temperature. It takes 23 days. It will only grow in seawater medium. Right? This is medium where they use seawater. So there's something else in the seawater that the organism needs. Don't know what it is yet. It has, again, a small genome. It's only 1.3 megabase pairs. It's a chemoheterotroph. And again, it has a rhodopsin-like molecule. This may again be why this organism can dominate in this environment, because it has this rhodopsin-like molecule that gives it more energy and an advantage over other microbes. Another organism that you find is Prochlorococcus. This is a very small cyanobacteria. It inhabits subtropical and tropical oceans, and it can grow at low light and deep depths. So it's very good at growing at deep depths where there's not a lot of light, and you'll see a lot of this in the ocean, as you saw in the survey. Finally, there's Syncococcus. This is found in all oceans. It's phylogenetically diverse, and you'll find it in fresh and ocean isolates. It is also an obligate photoautotroph, and it makes 0.6 to 1.7 micron ovoid cells. So again, this is a very common organism you find in the ocean. Okay, think back to our question about community physiology. What happens when you dump garbage into the ocean? Okay, if you think about this, what you'll realize is that the oceans have sulfate ion in them. Because they have sulfate ion in them, they're going to take polymers, degrade them, and then sulfate reducers will dominate, and you'll end up producing hydrogen sulfide. Hydrogen sulfide is a poisonous gas, and this has actually resulted in fish kills. So it's something we should not be doing. All right, our last topic in lakes and oceans is deep sea ocean vents. If you have not, go watch the videos on these amazing areas. Seeing what these things look like and learning about how they were discovered is amazing. I agree with the scientists in those videos. This is probably one of the most important biological discoveries of the 20th century. So what is a deep sea ocean vent? It's a physical environment that has volcanic activity between the tectonic plates. The temperature is between 5 and 400 degrees centigrade. The pH can be acidic to neutral, and water seeps into the cracks around this tectonic plate, this gap, and picks up hydrogen sulfide, iron, sulfur, ammonia, methane, and H2 from the magma. It then enters through the chimneys, and there's over 500 known systems at the bottom of the ocean. Here is the different things that get produced. All the seawater comes in, it gets superheated, it blasts out, it picks up all these ions, and you get all these reduced compounds being thrown into the ocean at these deep sea ocean vents. There is a huge amount of chemistry available, and you got it. Here's the electron tower. We've got a huge number of electron donors that can donate electrons to a whole bunch of electron acceptors. When that happens, and you have these electron donors and acceptors, you know there's going to be a huge collection of microorganisms and bacteria and archaea that can take advantage of them. So what do you think is available in the deep sea to use as a source of energy? It turns out you can have organic compounds and inorganic compounds. So what kind of microbes do you find in the deep sea? You find sulfide oxidizers. Chemistry is shown here. You find methanogens. Chemistry is shown here. You find methylotrophs that will use that methanogen and convert it into water. And you find hydrogen oxidizers. You'll note that there is oxygen present at the deep sea that they can take advantage of. One example of an organism that you'll find is Geolobus agangari. This is a eukaryota. It is a archaea, is a monococcus, and a thermophile. It can grow from 65 to 90 degrees centigrade, and it uses iron as its terminal electron acceptor and hydrogen gas as its electron donor. It can also use acetate and many other organics, so it's a very important microorganism in these environments and can take advantage of all sorts of compounds that are there. Another example of an organism that you find is Thermococcus atlanticus. This is a Euarchaeota. Again, it is an archaea. It is an obligate anaerobe. It is a thermophile. However, it is a heterotroph that grows on proteinaceous substrates. Some species of this genus can also use carbohydrates. It's of interest to biotechnology because it will grow in more mild conditions that are not as hard on fermenters, and then it has all sorts of thermophilic enzymes, and these are of great interest to industry. The last thing I want to look at here is the tube worms. Tube worms are an example of some of the macroorganisms that take advantage of the microorganisms that are growing there. And there's all sorts of life at these deep sea ocean vents that is not dependent on the sun. If you look at the anatomy of the worm, it has this tube and it will have a plume that comes out, and this plume absorbs CO2, hydrogen and hydrogen gas. Right? It brings those in and it pumps those into its body, and its body is full of microorganisms. These microorganisms are sulfur oxidizers, and they will take, right, so here's the sulfur oxidizers again. They will take hydrogen sulfide and oxygen and convert it to sulfate to make energy. They also take the CO2 and fix that into cell carbon. So the bacteria makes organic matter and then shares it with the tube worm. So in this mutualistic symbiosis, the tube worm collects the gases, brings them into its own body where these bacteria, these sulfur oxidizers are, and it provides them for it. And in return, the sulfur oxidizer gives them organic matter. Amazing collaboration and a completely foreign kind of organism. All right, that's it for lakes and oceans.